Maybe you can try the link now.
I have still not working.
All right.
It's 9 o'clock on my computer, but I don't know about your university.
Mine is also 9. Let me check.
Still the same message.
Yes, let's see. Okay. Okay, let me ask the assistant and maybe we can start.
We can wait till 1 minute past and try.
Okay. Oh yeah, maybe after 9:01. While waiting for student to come as well.
Yeah. Oh, 9:01. Let's try again. Nope, still the same message.
Okay, I will ask.
We'll do it at the end at 10 o'clock.
Okay.
5 to 10.
Shall I open this lecture now?
Yep.
Okay. Good afternoon, everyone. Thank you for attending this academic lecture by Dr. Philip Drake. So, Dr. Philip Drake is a lecturer in chemistry at the University of Bradford. And before working as a lecturer, he was working for Ministry of Defense in the UK for Defense Evaluation and Research Agency. And also after that, he began the career research as a researcher at the Industrial Technology Research Institute in Taiwan before become the lecturer at the University of. And his research interest is about polymer synthesis and characterization, nanoparticle, and also composite, nanoparticle composite, and also a sensor. Today, Dr. Drake will talk about the gold nanoparticles, about how to synthesize and the application. The screen is yours, Dr. Drake.
Okay, thank you, Ilma, for that introduction. Yes, so today I'm going to talk about gold nanoparticles, how we make them, what we can do with them, their shape, their structures, the chemistry. A brief introduction to myself, as Ilma said, my name is Dr. Drake. I work at the University of Bradford. I'm on Twitter, on X. If you want to follow me, feel free. That's my Twitter handle. I'm also on LinkedIn, so follow anything you like. I work in the area of material chemistry and nanoparticles. So one thing I do is make SIRS-based pathogen detection, so using a Raman laser system to detect bacteria using gold nanoparticles. I also work in the area of magnetic field hypothermia and cancer therapy using magnetic particles to treat cancer. So my work is involving a lot of nanotechnology. So basically, I make tiny probes, which are smaller than bacteria. We can then use those to find and to identify different bacteria using a laser. As the title suggests from my talk, I use gold nanoparticles to do this. It's all based on nanotechnology. So before we get into the details of nanoparticles, I want to just briefly introduce nanotechnology to you. It sounds like science fiction, making these tiny little things that can float around the body and detect bacteria and kill cancer cells. So nanotechnology in films is well known. First kind of inspiration that came to me was through a film called A Fantastic Voyage. This film was released in about 1966. I didn't see it at the cinema, I saw it on the TV. And it's about a little submarine which shrinks down into the body, and then the submarine drives around the body and tries to cure a stroke victim by destroying the plaque. There's a remake being made for the film, so it should be released in the cinemas at some point, but they've been talking about that for a long time. It hasn't happened yet. But essentially, they make a tiny submarine. Here's the submarine on the screen. It's about the same size as a red blood cell. It's called Proteus. Another film using nanotechnology would be
Recording in progress.
I did see this at the cinema. Might show you how old I am. But Inner Space is very similar. They make a little tiny submarine, shrink it down into the body. In this film, there's a good submarine and a bad submarine trying to kill each other inside the body. So the submarine floats around the body, finding obstacles and curing diseases. More recently, Star Trek, First Contact, Star Trek science is actually based on real physics. So a lot of the science in Star Trek is very close to what we think we could achieve. So in Star Trek, the Borg use the nanobots, these nanites. These are little tiny silicon-based robots which adhere to cells and work with cells and modify cells. So this is similar to something which we could, in theory, develop. And then we get into Ant-Man quantum realm. This isn't real. This is a little Ant-Man shrinking down into the size of, well, I don't know what it gets down to, subatomic levels into the quantum realm. It also appears in Endgame Avengers. Shrinking things down doesn't work. We can't do that. Consider the basic building blocks of what we're doing. The basic building blocks in chemistry are the elements, the atoms. In this case, let's look at a house. A house is made from bricks. This is a functional house. It's got doors, windows, and rooms. It has a purpose, it has a function, and it can be made from bricks. I estimate in this house there's about 15,000 bricks. We can make a smaller house from the same bricks. So the same building parts, we can make something a lot smaller. It's still functional. You could still live there as a door and a window. It's just a lot smaller. This smaller house, I reckon, has around 1,500 bricks. We then can make an even smaller house using the same bricks, the same building blocks. This house is no longer functional. It's a small house. You can recognize it as a house. However, it doesn't work. It's too small to live in. You could probably live in there if you were a cat or a dog, but it's too small for us. That house has around 150 bricks. So if you shrink the house down, eventually you lose functionality. If we go from a functional house, 15,000 bricks, to a non-functional house, 150 bricks, we can estimate the volume of those inside, and that's about 100 times smaller. So from 60 cubic meters down to 0.6 cubic meters, a shrinkage of 100 times. You lose functionality. If we look at the submarine in Fantastic Voyage, it sits around 3 or 4 people, so it should be around 20 cubic meters volume inside the submarine. You're shrinking that down to the size of a red blood cell. You're shrinking that down to about 4.5 times 10 to the minus 16 cubic meters of volume. That's a big number. You're shrinking by a lot. It's actually got a name. That's 40 quadrillion times. That doesn't work. You can't possibly shrink things down that much and expect them still to function the same way. So as objects get smaller, you need to start thinking about the size of the object compared to the basic building blocks. And we cannot change the basic building blocks. They're the atoms. So this is getting to the realms of nanotechnology. The atoms themselves. The elements. The periodic table. As chemists, you should all be familiar with the periodic table. It's what it looks like. I tend to use iron to make iron oxide nanoparticles. I use gadolinium. I dope the gadolinium into the iron oxide to make modified iron oxide particles. I use silica for glass coatings with oxygen. Silicon oxide is glass, so a lot of oxygen is in there. Carbon for the polymer coatings. There's always a lot of carbon involved. Carbon and hydrogen go hand in hand. Nitrogen's always around. We then get the elements of life, so the salts, potassium, calcium, magnesium, sodium are always present. Phosphorus in DNA is always there. And that's pretty much, combined with sulfur, all the elements I use. These are the basic building blocks of most of the work I do. Oh, let's not forget gold. Gold nanoparticles, the subject of this talk. So as things get smaller, the physical and chemical properties change. And that's the whole point of nanotechnology. As they get smaller, we change their physical and chemical properties. Gold is a really good example. Solid gold bars, you should all be familiar with gold. Very valuable, beautiful metal. Gold as we know it. If I zoomed in on the surface of this gold bar, you see gold. Nothing's changed. It's just gold. If you zoom in even further, using scanning probe technology, you can start to see the individual atoms. So this is a computer simulation of a gold surface where we can visualize atoms on the surface of the gold. You start to see the atoms. These are real pictures taken from a scanning tunneling microscope. 100 nanometer bar, we can see the individual gold lattice layers. If you zoom in closer, you can see the individual gold atoms. This is now a 1 nanometer bar, and these are individual atoms that we can see on the surface. If you keep breaking gold down into nanoparticles, you get little clumps of gold. So these are the gold atoms making up gold nanoparticles. We can start off very small. This one here has about 6 atoms. I wouldn't class this as a nanoparticle. I would class this as a gold cluster in chemistry terms. But once we get a bit bigger, we're definitely into the nanoparticle realm. These are no longer clusters, individual atoms. They're metal. Inside, they have metal particles, and they're just lumps of gold. So nanotechnology tends to be below 100 nanometers, a gold particle. This particle here has got lots of gold atoms in it. If it's only 2 nanometers across, it's still a tiny, tiny particle. This is about the limit of what we make in terms of nanoparticles. It has 2,000 gold atoms within it. We're going to get back into how we can estimate that and calculate that later in the talk. So we can make them different sizes. So these are TEM images of gold nanoparticles of different sizes. The smallest ones here are 5 nanometers. We can get bigger, 30 nanometers, 60, 100, 250. These are 400 nanometer particles of gold. As you change the size of the particles, you can change the color. So these small particles are the nice deep red color. The bigger particles become more a blue tinge, more of a purple pinky color. And simply by looking at the color of the solution, as a trained chemist, I can estimate the size of the particles. So 20 nanometers, you still have the nice red color. 60 nanometers, you start to get the pinky purpley color. In between, you get a mixture of the two colors coming together. So a 20 nanometer particle has around 250,000 gold atoms. So it's a lot of atoms in there. The 60 million nanometer particle has about 7 million atoms. So it is a lot of gold metal with a gold surface. So these are real images of particles which I've made. These are about 15 nanometers. There's a 20 nanometer bar here. These particles are fairly uniform in size. They're all around 15 to 20 nanometers. If we zoom in on the surface, we can actually see the lattice lines of the gold crystals. Bigger particles, again made by me, these are now 100 nanometer bars. So these particles are around 60 to 70 nanometers in size. And again, we get zoomed in on one single particle. So the gold clusters are actually lumps of gold atoms. We start down with the smallest, AU13. We're getting up to bigger sizes, AU147. This is a computer simulation of what different nanoparticles could look like with different numbers of gold atoms. It's about 0.6 nanometers. So these particles here are all less than 1 nanometer. This is 1.8 nanometers. These are still very small nanoparticles, about 2 nanometers in size. If we zoom in on a particle surface, we've got good resolution. You can see these black lines. These black lines are the crystal lattice lines. So these lines are the same as these lines you see within a crystal. It's the lines of the atoms lined up. So when we can see lattice lines within TM, it's a really nice thing to measure, determine the kind of crystal structure you've got. These are gold nanoparticles. We never see them like this in solution. These are bare surfaces. They're just gold atoms on the surface. That doesn't exist. You cannot have a bare atom on the surface. There's always a stabilizing ligand on the surface. And this is something people often forget. But nanoparticles have to have a stabilizing surface. So in reality, what you see is the gold atoms underneath, but then on the surface are the ligands. So if we zoom in on this surface, these are actually hexadecanoic acid, carbon 16. So there's 16 carbon atoms in a line ending in a carboxylic acid bonding to the nanoparticle surface. So these are the surfactant stabilizing ligands sticking off the surface. This gives the nanoparticle a very hydrophobic surface. So this particle will be soluble in organic solvent, not in aqueous solutions. And it's the chemistry of this ligand surface, of this stabilizing layer, which determines the chemistry of the particle. The gold has got nothing to do with the surface. You need to modify the stabilizing ligands to modify the chemistry. So all metal, whether it's gold, silver, copper, platinum, all metal nanoparticles need a stabilizing ligand on the surface. There's three ways we can stabilize them. One is by positive charge, one is by negative charge. We take a nanoparticle, we put a positive charge on the surface. All the particles have a positive charge. When two particles come together, the positive charges repel each other and the particles don't stick together, they get repelled and they stay stable in solution. So positive charge stabilization. Equally, we could have negative charge stabilization. All the particles have a negative charge. Again, when two particles come together, the negative charge repels each other and you get repulsion. Particles don't stick together, they stay as individual particles. We can also use surfactants, polymers, macromolecules, steric repulsion. So here we have a nanoparticle. We can put surfactants on the surface. These are like we just saw on the previous slide. This could be hexanechanoic acid, 16 carbons long, stabilizing the nanoparticle surface. Or we could have a polymer system. Now we've got a long chains of polymers all intertwined, all on the surface of the nanoparticle. The polymers repel each other, so when two particles come together, it's like a non-stick surface. They just bounce off each other and stay as individual particles. When they're made, gold nanoparticles are very stable. Here's an image of some gold particles. It's a glass vial. You can see the red color, characteristic of 10 to 20 nanometer particles. This sample is 170 years old and it's still stable. They're still well dispersed nanoparticles in solution. It was actually made by Michael Faraday back in 1850. You can see this particle in London. If you go to the Faraday Museum, you can see these on the shelf. Another example of stable particles would be the Lycras cup. This is the Roman cup using nanotechnology. It's made from a glassy material. It looks like a green glassy material on the outside. When light shines through it, you can see a red color appear. This is red from the nanoparticles, the gold nanoparticles within the ceramic glass. So nanotechnology isn't a new thing. The Romans were doing it several thousand years ago. So how do we make these particles? The most common method is described by Turkfish and friends. It's based on the reduction of tetrachloroaurate solution. It's initiated by sodium tricitrate in a boiling aqueous solution. So you take gold chloride, ionic gold, gold 3 plus, 4 chlorines gives you a minus charge on the ion with a proton stabilizing that. So it's gold 3, metallic, ionic gold 3 plus. You take trisodium citrate, citric acid, the sodium salts of that. We've got 3 carboxylate groups. This is a very strong reducing agent. So we take trisodium citrate, around 50 milligrams, dissolved in water, about a 1% solution. You add this to a refluxing solution of hydrogen tetrachloroaurate, 20 milligrams, in water. The drilled-in solution will go through a colour change. It'll go from being a yellowy, orangey colour to a deep red purple colour. You keep refluxing it. You then look at the UV vis spectrum, record from 300 to 900 nanometers, and you should see the surface plasma resonance peak, 517. We're going to come back to SPR peak later. It's a very important measurement. So the UV vis spectrum of a gold solution should have an SPR peak. Here we see the peak around 517 nanometers. And again, as a trained chemist, from that peak, you get a very easy estimate of the nanoparticle size. So what's happening? Gold chlorate, aurate, dissolved in water. Sodium citrate, They combine, it's a 2-electron reduction. The sodium citrate loses a carboxylate group and water, and it gives you gold 1. So you go from gold 3 to gold 1. It's still ionic gold, and CO2 is produced. Ionic gold comes together. 3 of the gold 1 ions come together with chlorine to produce 2 metallic gold atoms, and then 1 gold aurate again. So this is a cluster of just 2 atoms coming together. They're gold 0, which means they're now metallic gold. So the nucleation site, you get your gold clusters, 2 atoms together, very small. The rest is still gold 3 and the citrate. That keeps on reacting to start building the nanoparticles up. So you grow the particles. You go from a small cluster of 2 or 3 atoms to final nanoparticles. These are less than 1 nanometer. These are around 13 nanometers as determined by the SPR peak. These clusters are unstable. You can't see them, you can't isolate them. They're theoretical. The gold nanoparticles is what you can see and you can characterize. So particle synthesis is made from nucleation and then growth. And this is the key to all nanoparticle production, controlling the nucleation and controlling the growth. That determines the size and shape of your particles. If we have less nucleation sites, same amount of gold nanoparticle, same amount of gold metal, we're going to get bigger particles. All the gold will form just 3 particles, 3 big particles. If we have more nucleation sites, we're going to get less gold because the same amount of gold will now coat all these, giving you more nanoparticles of a smaller size. If we have uncontrolled nucleation, so we have nucleation and growth all happening at the same time, we get nucleation site happening, that grows to a nanoparticle with more nucleation sites, more growth, you get a mixture of different particle sizes. So the best way to get homogeneous particles is to have controlled rapid nucleation followed by controlled growth. If you get heterogeneous particles, a very big size distribution is when you have growth and nucleation all occurring at the same time. We can even make larger particles. So using the Crooked-Fakes method, the biggest particles you can make are about 20 nanometers. No matter what you do, you're going to get the red particles about 20 nanometers in size. To make bigger particles, you need a different method. You use seed particle growth. So in seed particle growth, you take your gold chloroaurate, mix it with some pre-made gold nanoparticles of a known size. You then add in the sodium citrate, and you start to get growth on those particles again, which gives you a blue color after about 30 seconds. And now the SPR peak has shifted. It's gone from 517 to about 535. So I now know that these particles are about 51 nanometers. So the SPR peak is the way to record the nanoparticle size. So you take your pre-made particles, about 13 nanometers, you add in some more citrate and some more gold chloride, and they can grow to give you larger particles. Now, there's no nucleation now because we're using these as the nucleation sites. The more of these we add, the smaller the particles, the less of these, the bigger the particles. So we can control the size of the final particle by controlling the amount of nucleation sites or the amount of gold added. We can aim very precise sizes. And this can make particles from 20 to 100 nanometers in size. So, particles, small, nice deep red color, big, more a violet color. This is in transmission mode. This is when the light is coming through the sample towards your eyes. As you get these larger particles, you start to get scattering. So small particles, 20 nanometers in size, a red color, no scattering. As the particles get bigger, the light can hit the particles and get bounced off towards you. So now we're seeing scattering. The solution is no longer transparent. It has scattered light as well as transmitted light. So using the citrate method, you can make hydrophilic, water-soluble gold particles from 10 to 100 nanometers. You can very carefully control the size. You can make different sizes, and you can see the different properties. That's in transmittant, that's in reflectance mode. These kind of particles are stable for years. We've seen Faraday's particles, they're almost 200 years old and still not settled. The bigger particles start to precipitate, they'll slowly start to settle down. So after a few weeks, these will show sedimentation, solution will become clear, and you'll have a film on the bottom. You can remix them and reshake them very easily, but they do settle out over time. Bigger particles here, these are now about 60 to 80. These settle in a few days. And then the bigger particles, 80 to 100 nanometers, they can settle out within a few hours. You can see them precipitating straight away. You can always redisperse them and shake them. So that's the settling time. The AU nanoparticles show a surface plasmon resonance. This gives rise to the color that we see. The SPR peak changes depending on the size of the particles. So the small particles around 500 nanometers, bigger particles about 550 nanometers. And you can plot out the SPR peak maximum as a function of particle diameter. And there's a very nice linear fit. So if you measure the SPR peak maximum, you can read off the size of your nanoparticles. The SPR peak comes from the electrons on the particle surface. So it's metallic particles are in metallic conduction band. As the light passes over, the wavelength of light passes over, the electron cloud can be distorted above and below the particle. That's the transition you're measuring when you're looking at the SPR peak. So those were hydrophilic particles. These are all dissolved in water. We can also make particles hydrophobic, so we can make them dissolved in organic solvents. So to do that, we use the Brust method. So Brust first described in 1994, it's a two-phase synthesis process. It can make very small gold particles, AU nanoparticles of about 1.5 nanometers to 6 nanometers, soluble in organic solvents. You take a surfactant, tetrahexammonium bromide in this case, this is our surfactant, that's dissolved in toluene. You add in hexane thiol, we'll talk about that in a minute as to why that's there. You add that to solution. You combine that with an aqueous phase gold tetrachloroaurate. So it's the same source of gold, hydrogen tetrachloroaurate in water. This gives you two phases, toluene phase and aqueous phase. Shake them and mix them, you'll see the tetrachloroaurate transfers from aqueous into the toluene phase. You then add in a reducing agent, sodium borohydride. This is what reduces the gold 3 to gold 0. You see a color change. Once you add the sodium borohydride, you can see the color change from an orange solution to a deep ruby red, typical of gold nanoparticles. So the Brust method for reduction of gold is conducted in organic phase using sodium borohydride. You add sodium borohydride as a strong reducing agent. This gives you very quick nucleation. So we have lots of nucleation sites occurring very quickly, which means we get very small nanoparticles. The thiol, remember we added hexane thiol. The thiol is a capping agent that reacts very quickly with metallic gold and forms a very stable bond with gold. So as soon as the nanoparticles start to form with gold 0, the thiol bonds to the surface and stabilizes them. So it forms like a covalent link between the gold and the sulfur, and it's impossible to remove. Once the thiol bond forms on gold, it's very difficult to remove it. So this gives you very small nanoparticles, 1.2 nanometers, the smallest ones you can make. They have an SPR peak at about 510 nanometers. We can do the same synthesis, but we can add the thiol later. If we add the thiol after the sodium borohydride, you make the particles and you then grow them into bigger particles and then add your capping agent. You can get about 4.8 nanometer particles. Still small, you still have sodium borohydride, there's still lots of nucleation, so you get small particles. So by using a thiol, we can control very precisely the size of the particles, either having it in the beginning of the reaction or adding it at the end of the reaction. And these are organic solvents. So these are soluble in toluene, in chloroform, in hexane. So 1.2 nanometer particles. We're getting into this kind of range here, AU57, AU55. Very small. The surface is stabilized by the thiol. So this is the hexane thiol. The sulfur and the gold forms a covalent bond. This is your gold atom. This is your sulfur atom. There's a very strong bond, and then you have a hydrophobic surface. So what you make is your gold cluster, very small, stabilized with the surfactants all around the surface. So what you see is the organic phase. You see the carbon phase on the surface. An example of some small particles. This is a 20 nanometer bar. These are the small particles you can make. How do we measure the size? We can use SPR peaks and UV-Vis. The best is using TEM. TEM, we physically measure the size of the particle. We know the scale bar, we measure the size of the particle. We can get a ruler out, measure it. We can get a whole list of particle sizes. Just measure lots of particles using software or using a ruler, list them all down. Once you get a whole range of particles, you can then take the average and you can work out the standard deviation. So from these particles, we have an average size of 4.79 nanometers with a standard deviation of 0.77. This is what we need to characterize our particles, the average size and the standard deviation. We could do that with bigger particles too. So here we have 50 nanometer particles about. If we take lots of particles, we can measure the particle size using a ruler or using software. We can put out a normal distribution curve, and we can look at the frequency distribution with the points. So the histogram is our frequency distribution. We look at bin sizes and plot out how many particles within that range. And the black line is the normal distribution fitted to an average and a standard deviation. Once you know an average and a standard deviation, you can plot the normal distribution and make sure that fits to your data. It should be a bell-shaped curve. Not all particles do the same kind of curve. So here we have a histogram as kind of fits. We can see lots of different types of histograms. This one, I wouldn't want to fit this to a standard deviation, a standard normal distribution. It's not enough data. So you should always show a histogram. You should always show the average and the standard deviation. And hopefully they always match nicely, but they don't always match. So do think about how your distributions are affected. So you now know how much gold you've added. You know how many gold ions you added. You assume 100% of this goes into your nanoparticles. So you can give a mass concentration. This nanoparticle solution has a mass concentration of 0.1 grams per mil of gold. If you want to know the molar concentration, you need to know how many gold atoms are in your nanoparticles. You need to know the average size of your particles. You can estimate this from TEM or from UV-Vis spectrum. If we know the size, we can work out the volume of a sphere. So once you know a diameter or a radius, you can work out the volume of a nanoparticle, 4/3 pi r cubed. We know the size of the gold atom, AU 0.288 nanometers in diameter. So if we know the volume of this, and we know the volume of our big sphere, we can work out how many of these are in the big sphere. Particles form hexagonal close packing. So there's about a 74% efficiency. So there's 26% of the volume has nothing in it. So work out the volume of your big sphere, times it by 0.74, gives you the volume of your gold. Divide that by the volume of one gold nanoparticle, gives you how many particles are in the nanoparticle, the system of sphere. And then we can plot them out. We can plot two numbers. The blue line is total number of atoms in the nanoparticle. The other line is surface atoms. So what's important with nanoparticles is the ratio of surface atoms to core atoms. As you get smaller, the number of atoms decreases, the number of surface atoms becomes closer and closer to normal. So a 10 nanometer particle has about 35,000 atoms in its core. It has about 5,000 on the surface. And as we get down to 1 nanometer, you can see at 1 nanometer, all the atoms are surface atoms. 1.5 nanometers, you've got some in the core and a lot on the surface. And it's ratio of the surface to core, which is important. So 0.86 nanometer particle is about here somewhere. All these are surface atoms. With the bigger particles, you've got core atoms which are masked and the surface atoms involved in the chemistry. So the big particles, we know there's about 4 million gold atoms and about 132,000 surface atoms. Small nanoparticles, we know there's about 4,000 atoms and about 1,000 surface atoms. There's a strong color dependence, and this is what makes gold nanoparticles very interesting, is the color change from red to blue. Just by looking at the solution, you can estimate the size. So your eyes can become sensors to work out the size of nanoparticles just by looking at the color of solutions. But it's not just the size of nanoparticles, it's how they're aggregated. So if the nanoparticles are touching, they become blue. If they're individual particles, they stay red. So in 2006, Chad Merkins used this color change to develop a DNA sensor. To do that, he took DNA onto the surface of a gold nanoparticle. So you take your gold particle, you have DNA on the surface. It's the same DNA on all the surface, all the particles. So the particles don't interact. They bounce off each other. This DNA doesn't interact with the other DNA. If you add in the complementary strand, so this is now the DNA you want to detect. This is a complementary strand. It binds to both of these. So the DNA forms a double helix on one particle, leaving a bit sticking off. This forms a double helix with a second particle, so the particles now become coupled together. When they do that, the color changes. It goes from being red to being blue. So now you've got a sensor that detects this particular strand of DNA, this sequence of DNA. If it's present, you get a blue color. So this is a way of developing the first kind of sensor for DNA. We can also use lateral flow assays. So a lateral flow assay, you look for a capture antibody for viruses. That's the capture antibody, put that on the surface. You take some virus protein or DNA. There's the virus protein on the surface. You have a gold nanoparticle labeled IgG. So this is chicken IgG with a gold nanoparticle stuck to the antibody. that can float down the lateral flow system. Put a blood on the surface, flows down. That's got COVID antibodies in there. The COVID antibodies recognizes the COVID protein. The labeled antibody recognizes the human IgG, and you get 2 lines appearing. These lines are labeled using gold nanoparticles. If there's no COVID present, IgG present, you only get 1 line. If it is present, you get 2 lines. So positive, 2 lines, negative, 1 line, all visualized using gold nanoparticles. And the whole world is now very familiar with this kind of lateral flow system since the COVID pandemic. But that's how it works. Think about the size of the particles. We've got antibodies and we've got gold nanoparticles. How big is a gold nanoparticle compared to an antibody? Here's some computer simulations. This is a 20 nanometer gold particle. This is an antibody. They're about the same size. If we get down to iron oxide particles, they'll be a bit smaller. Single peptide proteins are a bit smaller still, small molecules. So gold nanoparticles are about the same size as an antibody. So gold can undergo ligand exchange with thiol compounds. The captobenzoic acid binds to the gold surface very well and gives very stable gold particles. Anything with a thiol in will always bond to gold nanoparticles. A captobenzoic acid, a benzene ring, carboxylic acid group, and a thiol. Mix that with gold nanoparticles, you get a negative charged surface, very well stabilized, with a sulfur attached to the gold. This has a very strong Raman signal, and because of the nanoparticle, you get a very strong surface enhanced resonance Raman. So when your Raman laser matches the SPR laser of the gold particles, you get enhancement and you get very strong signals. These 2 peaks are characteristic of the protons on the benzene ring. The carboxylate surface is the peak in the middle. So using mercaptobenzoic acid nanoparticles, labeled nanoparticles, we can make SIRS systems which can detect femtomole levels. So we can now count nanoparticles at a very low level, 1 times 10 to the minus 21 moles. And then we can use these as labels. When we shine a Raman laser on the system, we get these 2 peaks appearing. We can detect these nanoparticles very easily. So we can label antibodies using these nanoparticles. And that gives you ultra-sensitive detection systems. So typical example, using mercaptobenzoic acid labeled gold particles, we can mix them with mouse IgG. We can then get a correlation chart which links the concentration of the IgG to the nanoparticles. So using a whole different range, this is just a standard method of using mouse, anti-mouse IgG, detecting rabbit IgG. So we take our goat-coated gold nanoparticles with some anti-mouse IgG on the surface. So these are our capture lines. This is rabbit IgG that detects mouse IgG. We add in the mouse IgG. It floats on the surface, it sticks to certain points. We then take our gold nanoparticle labels, which recognize mouse IgG. So they stick to the mouse IgG on the surface. We can wash off anything that's not bound, gets washed off. We then take the Raman laser and shine it on the surface. These nanoparticles will scatter the light and give us our signal. And you get a very nice linear dependence concentration of mouse IgG in the picomolar region. So we can actually detect femtomole detection. So we can detect a very, very small amount of IgG using laser systems. We can also use this to detect bacteria. So Bacteria detection is really important in many diverse areas, hospital settings, in clinics, in homes, for national defence and bioterrorism. But there's a clear barrier in the current detection limits and what we want to be able to do. To detect bacteria, we tend to have colony counting and culturing. So we need to grow them on a plate for a few hours or even a few days. We can detect about 150,000 bacteria cells. But we need to be able to detect one or two. Eating 10 E. coli can give you food poisoning, so just 10 bacteria cells. So we need better methods of detecting bacteria, and gold nanoparticles offer that option. So the Staphylococcal aureus, it's a kind of bacteria. You have a blood concentration of more than 50 CFUs per mil, then you have an infection. You need to be treated very seriously. E. coli 0157 causes an infection with just 10 CFU. CFU is a bacteria colony forming unit. It's how you count bacteria in medical terms. So for dairy collection, collecting milk, you've got to show you've got S. aureus in milk is less than 10 CFU per mil. And yet our detection limit is around 150,000 CFU per mil in 12 to 24 hours. That's using PCR and SPR based methods. So can we do better? Well, yes, we can. This is the concept. We take S. aureus, we take our mercaptobenzoic acid labeled gold. We also take some iron oxide nanoparticles and some single domain antibodies. S. aureus has protein A on the surface. Our gold nanoparticles, labeled with single domain antibodies, and iron oxide particles labeled with single domain antibodies, bind to the surface of S. aureus. So you get gold particles and iron oxide particles on the surface of the bacteria. So now the bacteria is magnetically responsive from the nanoparticles and gives you the strong signal from the gold SIRS detection method. So we take a laser system, we take a solution with mixed things in there, put a magnet on the surface. Anything with magnetic particles gets attracted to the magnet. Anything with no magnetic labels sinks to the bottom. You then shine your laser onto where the magnet is. The gold particles attached to the bacteria gives you a strong signal back to your Raman laser. So this is how the system works. This is the laser mounted within the cavity. The laser is focused on the end of a tube, and this is magnetic trapping, where the magnetic particles will get trapped by the magnet, and that's where the laser focuses on. This is a SEM of gold of Staphylococcal aureus. This is the bacteria, the round bacteria. That's the surface zoomed in. If we mix it with the gold particles, we can see the gold particles on the surface. So these bright spots are the gold nanoparticles bound to S. aureus. So that's one of the nanoparticles on the surface there. We can also confirm that by EDX characterization. We get a little gold peak saying gold metal is there. The SIRS measurement, those particles give us those two characteristic peaks. And we can link the peak height to the concentration. So what we get is a linear dependence between SIRS peak height and the concentration of S. aureus. SA's S. aureus, different concentrations. So with the gold particles, we can detect 2 CFU per mil of S. aureus. Standard PCR can detect 2 million. If you culture the bacteria for 24 hours and run PCR, you can detect 20 CFU, but even then you can't detect 2. With the gold nanoparticles, you can detect all the way down to 2 CFU per mil. You can also combine the gold nanoparticles into a DNA detection method similar to Merkin's. So DNA is a double helix. You have a single strand. You then have the complementary strand, forms a double helix. In PCR, you take your target DNA, you denature this, so you split these two DNA strands apart to single-stranded DNA. You then add in a primer, 2 primers, one for each strand. The primers bind to the DNA strands. Once the primers bind, you add in a polymerase. It's an enzyme. It detects the primer and then extends and rebuilds the DNA chain. So extension occurs, and you get two strands of DNA from one double helix to two double helix. You then repeat, denature these, and go through the cycles. This is how PCR works. So we can use the same iron oxide particles and the same gold nanoparticles, label them with the primers on the surface, and we get a system where we can detect DNA. So we take our iron oxide nanoparticle with one of the primers on the surface. Our gold nanoparticle has the other primer. These don't combine together, so they don't interact. They're stable in solution. You add in the DNA you want to detect. When you add it in, that forms a double-stranded DNA to the gold, to the iron oxide particle. Polymerase extension occurs. So then the polymerase comes in, it extends that particle, extends that DNA. Once extensions occurred, the gold nanoparticle can then bind in. So now the gold particle binds in, and you get a complex forming between the gold and the iron oxide. This gives you a magnetic SIRS response depending on the concentration of the target DNA. And using that system, again, we see the same two peaks. It's still mercaptobenzoic acid labeled gold. And we can link these peak heights to concentration of DNA, where you get a linear dependence between SIRS peak intensity and DNA concentration. Using this method, we can detect down to 10 attomolars. That's about 3,000 times more sensitive than PCR. So current standard art is PCR methods. This is about 3,000 times more sensitive because of the SIRS from the gold particles. So you can also use gold particles for other things. You can use them for treating cancer. There's a photothermal effect. So gold particles don't have to be round. You can make them different sizes. You can make them round, squares, lots of little spiky surfaces, nanotubes. When you change the surface of the gold, the shape of the gold, you change the SPR resonance, and you can make it shift down to the infrared region. So standard gold particles have the SPR peak around 500 to 600 nanometers. As you make nanorods, you get a new SPR peak occurring down at 900 nanometers. So this is now in the near infrared region and becomes a very strong absorber. So we can use a near infrared laser to excite this resonance peak within the living structures, and that produces thermal heat within inside tumors. You can also use it for drug delivery. So a gold nanoparticle, as we saw, was big. Because it's big, you've got lots of options for adding different things to the surface. So you can add in a drug molecule. You can add in targeting ligands. You can add on DNA and RNA. So you can modify the surface of gold nanoparticles with lots of different components all at the same time. So you can make very good drug delivery systems where the nanoparticle carries a high concentration of drugs into a cell, and you can then have some kind of mechanism where the drug is then released from the particle within the cell. So they're very good for thermal therapy. They're also very good for drug delivery systems. Because they're big, we've got thousands of surface atoms. We can imagine modifying this with lots of different antibodies. We can get 4 or 5 different antibodies on the surface of one gold nanoparticle, or we can get lots of drug molecules on the surface, which can be released when we want them to. So that brings me to the end of the talk. Gold nanoparticles are very useful things. We can do lots of things with them. We can use them for sensors. We can use them for drug delivery. We can use them for therapy. Any questions?
Thank you, Dr. Dick, for the explanation about the gold nanoparticles and application. Is there any question from students? There may be, there is a colleague here, Dr. Della. Oh, okay. Oh, yeah. There is one question in the chat box From Igusti Agung Setiawan. Excuse me, sir. Let me introduce myself. My name is Igusti Agung Setiawan. I would like to ask if cold nanoparticle can influence the light spectrum. Could this method be used to treat the retina of blind individuals, or maybe can be increase human vision like superhuman?
Can nanoparticles influence the light spectrum? Yeah, so we looked at how we can change the SPR peak. The SPR peak is how the nanoparticles absorb light, and we can shift that to different regions. So we can have it in the colored region, so in the absorbing the blue region, so we get red particles, or we can shift it to the near infrared region. Can influence the light spectrum. Yeah, could this method be used to treat the retina of blind individuals, or maybe can be increased human vision like superhumans? I don't think you'd use nanoparticles to do that. We can do that kind of work. So you can have glasses. You can imagine Google glasses where you can start to see different spectrum. So that's a whole different area of research, is looking at how we see the visible spectrum, colored light. But you can develop sensors that detect the infrared region, thermal cameras, or you can detect the UV vis region. So we can have different goggles, different glasses to see different spectrum, different wavelengths. I wouldn't use nanoparticles to do that.
Okay, thank you. Is there any question? Okay, just reminder for student, don't forget to fill the attendance form.
Yeah, I
Please.
have to do that myself.
Oh yeah. Yeah. Okay. Is there any more questions? I would like to ask Dr. Drake. So you said that for the spheric gold nanoparticles, and if they are having a bigger size, the UV spectra will shift to the right to bigger wavelength number.
Yes.
So is it always happen in every nanoparticles, or is just like there are like a specific for gold nanoparticles, spheric one, or because when you show us the